Hello, everyone. My name is Chelsey Skeete. I am a fifth-year PhD student at Ram Gummilo's lab, and today I'll be discussing opioid signaling enhances microglial HIV infection and virus-induced inflammatory responses. So currently, there's a syndemic of people who are chronically using opioids and living with HIV, which is estimated to be about 3 million people on a global scale. These populations of people are at higher risk for various comorbidities, including HAND, which stands for HIV-associated neurocognitive disorders. This is a broad spectrum of neurocognitive deficits marked by chronic inflammation within the brain. It can range from asymptomatic to mild, all the way to dementia forms. In this study, we are focusing on microglia as they are key regulators of neuroinflammation, and they are the main reservoir of HIV in the brain. Upon infection, they transition to this pro-inflammatory state in which they secrete uh, inflammatory cytokines and chemokines that are suspected to damage neurons in surrounding areas. Now, there's a higher incidence of HAN in people who are chronically using opioids, even if they're on antiretroviral treatments to suppress viral loads. Additionally, antiretroviral interruption is higher in people who chronically inject drugs. So it's important to understand these mechanisms that may contribute to viral rebound. So just to give you a general sense of the viral life cycle, after entry, viral encoding occurs and capsid then enters the nucleus in which viral RNA is transcribed into DNA. This then integrates within the host genome, which is termed as the provirus, and is transcribed under NF-kappa B activation, which then triggers RNA to be spliced, translated, and virions are secreted out of the cell. Now, RNA can also be unspliced, in which intron-containing RNA can be exported out of this CREM1 pathway, and this can trigger viral sensors such as MDA5 and MAVs to trigger these innate immune responses that are downstream of this IRF pathway. Now, our lab has shown that HIV infection in this pink bar induces IP10 and other innate immune responses. And this response can be blocked using verdinexor, which prevents intron-containing export out of the nucleus. So we're hypothesizing that intron-containing RNA expression induces innate immune responses in microglia. Now, how does this connect to opioid signaling? Well, just to provide you with a brief overview of opioid signaling, opioid receptors are classified as GPCRs, in which mu is the most abundant opioid receptor within the brain. And mu can be activated by many different opioids, including morphine, and also blocked by antagonists such as naloxone, which is also referred to as Narcan, used to reverse overdoses. And so upon activation of mu, the receptor is phosphorylated, which triggers a wide range of phosphorylation cascades. Only one of those cascades we're focusing on today, known as the PI3K AKT pathway, which has been shown to induce NF-kappa B activation. So we're hypothesizing that both opioid signaling synergizes with HIV infection to promote HIV infection within microglia and also promote innate immune responses. And again, we're focusing on microglia because they are permissive to infection. They regulate these innate immune responses and express mu. So our main question is, what is the effect of morphine on HIV infection and microglia? And we're hypothesizing that morphine will enhance infection at various stages of the replication cycle. So just to give you a sense of the different cell models we are using, the first cell model is known as iPSC-derived microglia. This is in collaboration with Gustavo Mostoslavsky at the Center of Regenerative Medicine here at BU, where we confirmed that mu is expressed here in this green staining and overlaps with plasma membrane staining here in red. And so we confirmed that mu expression is on the surface of these cells. However, monocyte-derived macrophages lack this green staining, showing that they are deficient for mu expression. So we use macrophages as a negative control for mu expression. We also use this THP cell line, which is a monocyte cell line that can be differentiated into macrophages using PMA. Controlled THPs do not express mu, as you can see with this lack of green staining here. However, when we overexpress mu in THPs, we can now see the staining that co-localizes with the plasma membrane stain. And we use these THPs as a rescue model to rescue any effects of morphine on HIV infection in macrophages. Now, for our study, what we did is we pre-treated cells with either an opioid antagonist known as naloxone, a PI3K inhibitor known as warmantinin, before the addition of morphine treatment for 24 hours. Cells were then infected with replication-competent HIV and then harvested 3 days late post-infection for DNA, in which we measured pro-virus establishment and reverse transcripts, RNA, in which we measured full-length RNA that is unspliced, and supernatant, in which we measured cytokine secretion and virion release. We confirmed that these receptors overexpressed in THPs are functional by showing that morphine treatment here induces phosphorylation of this mu opioid receptor, which is blocked by naloxone treatment. And this phenomenon is not observed in controlled THPs. We then asked, does morphine treatment induce phosphorylation of AKT during HIV infection using Western blot here, in which we quantified phospho-AKT here in this top band to the total amount of AKT and also to the loading control in which we use beta-actin. And what we saw is that morphine treatment had no significant impact on phospho-AKT in controlled THPs. However, when mu is overexpressed, morphine does enhance phosphorylation of AKT, which is blocked by naloxone and warmantinin. So what we see is that HIV infection and mu activation does synergize at this PI3K AKT pathway. Next, we looked at reverse transcription, looking at earlier steps of the replication cycle within HIV. And in macrophages, we see that morphine had no impact on reverse transcripts detected by PCR. However, morphine treatment does significantly enhance reverse transcripts when mu is exogenously overexpressed, and this is blocked by naloxone treatment. The next step after reverse transcription is proviral establishment, which is again that viral DNA that's integrating with the host genome. This was detected using digital droplet PCR in collaboration with the Henderson lab. What we saw is that morphine treatment does enhance intact proviral establishment in microglia, which is blocked by naloxone, and this was not observed in the macrophages. However, when mu is overexpressed in macrophages, we see that morphine does enhance intact proviral establishment by twofold, which is blocked by naloxone. The next step after proviral establishment is transcription, in which we also measured full-length intron-containing RNA using digital droplet PCR. And we saw that morphine enhances intact full-length intron-containing RNA by fourfold here, which is blocked by naloxone treatment here in microglia. However, macrophages, we see that there's no impact of morphine treatment on intron-containing RNA expression. However, when mu is overexpressed in these cells, we see that morphine now enhances intron-containing RNA similar as we observed in the microglia. Finally, we looked at that last step of the replication cycle, looking at virion release. This was measured by using a P24 ELISA to measure the release of capsid protein known as P24 GAG. And what we see here is that, again, morphine does significantly enhance P24 secretion in microglia, which is blocked by naloxone. This phenomenon was not significantly observed in macrophages. However, when mu is overexpressed, morphine does enhance P24 release. So what we conclude from this is that morphine does enhance HIV infection in a mu opioid receptor dependent manner in microglia. And so our next question was, what's the effect of morphine on HIV-1 induced innate immune responses. We're hypothesizing that morphine would enhance these responses as infection is enhanced in these cells. And so what we did was we saw that morphine treatment does enhance IP-10 secretion and microglia, shown here, which was only observed in the context of infection, and this was blocked by naloxone and by wart-mannanin treatment. We do not observe this effect in controlled THPs shown here. However, when mu is exogenously expressed in macrophages, we see that morphine now has an impact of enhancing IP-10 secretion only in the context of infection. So from this data, we conclude that microglia express mu while macrophages have deficient expression. Mu is necessary to observe morphine-mediated enhancement of infection at various steps of the replication cycle, and mu activation by morphine enhances intron-containing RNA, which correlates with IP-10 secretion. And the use of these opioid antagonists and PI3K inhibitors block morphine enhancement of virion release and IP-10 secretion. With that, I would like to thank my lab, our collaborators in the Gustavo Moslowski's lab, and also in the Henderson lab. And I would like to thank the Clinical Translational Science Institute for this opportunity. Thank you.